friends, good afternoon, good evening. In this video, today I'm going to be talking about cells. I'm going to be discussing the different types of cells, the important structures and functions inside of the cell. Okay? All right, we'll get started right away. In our textbooks, this is going to be chapter 6, or it's going to be titled A Tour of the Cell. So what's going on inside of the cell? But before we talk about what goes on inside of the cell, let's talk about the two main categories or the two types of cells. Oh, before that, we need to go over the objectives, okay? The objectives in this part of our unit. So for this unit, you must know the three differences between prokaryotic and eukaryotic cells, the structure and function of organelles common to plants and animal cells, the structure and function of organelles found only in plant cells or only in animal cells. All right, so there's two types of cells that you can find on our planet. You first have prokaryotes, which are domain bacteria and archaea, and then you have the eukarya or eukaryotes, which are found in domain eukarya, which include protists, fungi, plants, and animals. Okay, we have basically, there's three major domains of living organisms. You have bacteria, archaea, and eukarya. Those are like the three domains, the three groups, I guess you can say, like the three big groups of organisms found on our planet. You have all heard of bacteria before. You may not have heard of archaea, but archaea are a type of prokaryote that live in extreme environments. So oftentimes we refer to them as the extremophiles. Okay, like these are the organisms that live in extreme environments, like deep sea vents, or in extremely hot environments like geysers. Okay, or these could be organisms that live in extreme pHs. All right, these are organisms that cannot, that other organisms, they live in environments where other organisms could never survive in. Okay, and we humans, we belong to domain eukarya because our cells are eukaryotic. Okay, and we belong to kingdom Animalia. So these are four different kingdoms. You got protists, fungi, plants, and animals that are under the large category of eukarya. All right, so let's talk about the differences between prokaryotes and eukaryotes. Oh, before that though, okay, so these are the three domains that I was referring to. You've got the bacteria, archaea, and eukarya, and here we are. We belong to kingdom Animalia. There's other kingdoms like fungi, plants, okay, ciliates, flagellates, slime molds. Those are actually, they belong to Protista, okay, kingdom Protista. But these are the different kinds of bacteria, and then archaea. All right, so this is what a typical prokaryotic cell or bacteria looks like. So when you hear prokaryotic, you should think bacteria, okay? And bacteria have very, very simple structures. There's not a lot going on inside of bacterial cells. They're very simple, and that's why they can reproduce so quickly, is because, you know, their internal structure is very simple. It can easily be replicated, and they can reproduce like within 20 minutes, okay? Their lifespan is only 20 minutes. And inside of the bacterial cell or prokaryotic cell, you see DNA. There's no nucleus. The DNA is kept inside of the cytosol. The cytosol is basically the fluid inside of the cell. You have a cell membrane, and you might have a cell wall, and then you might have a capsule. Okay, and sometimes they can move around through flagella, okay, move around using flagella, which are like tails. Okay, very simple. Oh, and I forgot to mention, they do have something that we have in our cells, which are ribosomes. These are the little ribosomes, and those ribosomes are going to synthesize, make proteins. Okay, so that's the structure of a prokaryotic cell. So when we compare prokaryotes versus eukaryotes, that word pro means before, karyo refers to kernel or like grain, okay? And, you know, back in the, you know, early scientific days, okay, when they were looking at cells under a microscope, they noticed that with like human cells, they could see some kind of grain in the middle of the cell, okay, the kernel. The karyo refers to the kernel, like a little grain, like a little dot inside the cell. And so that dot turned out to be a nucleus. So eukaryotes, we have a nucleus. Whereas a prokaryote has no nucleus. So when we say prokaryote, that means before a nucleus, before the nucleus came into being, before it evolved, okay? Whereas eukaryote, we have a true kernel, we have a true nucleus. You can see that grain, okay? So the number one difference between prokaryotes and eukaryotes is when you look at bacteria, so let's say that's a bacteria, there's no nucleus, there's no kernel or grain that you can see, whereas in our cells, you can see a little something in the middle. That is the nucleus, that's the grain, okay? So we have a nucleus, and that nucleus, there's an envelope that protects, envelope meaning membrane, there's a membrane that protects our DNA because our DNA is inside of there. Whereas with bacteria, their DNA is like floating in the cytosol, and that area where you can find the DNA floating is, we just call it a nucleoid, okay? We have a nucleus, they have a nucleoid, okay? We have an envelope that surrounds our DNA, and that envelope is a membrane or the nuclear membrane. They don't have a membrane that surrounds our DNA. So cytosol is that fluid. We have cytosol, and bacteria have cytosol. They have no organelles other than ribosomes that are going to make proteins. We have many membrane-bound organelles that have very specific structures and specific jobs, specific functions, and we're going to be going over that today in this video. Prokaryotes are much smaller than eukaryotes. So eukaryotes are much larger in size. And when I say primitive, meaning that they've been around on our planet for a long, long time. They're very simple, whereas eukaryotes are much more complex in structure. So examples of prokaryotes would be bacteria, archaea, whereas examples of eukarya would be like plants and animals, okay? Okay, the next topic is that cells must be small. You can only see them with the microscope. And the reason why is that cells, they cannot survive, they cannot live unless they have a large surface area to volume ratio. Okay, so in math, you probably have learned these equations. For a sphere or a cube, surface area is 4 pi r squared. For a cube, it's going to be 6s squared. That's the 6 sides, right? And then the volume is 4/3 times the pi times the radius to the 3rd power, okay? Volume of a cube is going to be the surface area squared, okay? All right, so large surface area allows for an increase in rates of chemical exchange between cell and the environment. So what I mean by that is that let's compare a small cell versus a large cell, okay? If you have like food, okay, just outside of the cell, and that food is going to be absorbed by a small cell or by the large cell, that food is going to feed the entire volume of this small cell a lot faster than the volume of a larger cell, okay? So a large surface area to volume ratio, it makes it much more efficient for the cell to exchange chemicals coming in and also waste going out, okay? So let me just erase this so that we can, because I'm going to be adding more to this slide. Oops. I guess I could have just erased the entire thing. Okay. So again, this is showing you that when you compare a small cell with, like, let's say the side is 1 millimeter versus a larger cell, which is like 5 millimeters, if you do the surface area of this cube, it's going to be 6. Surface area of this larger cube, it's going to be 150. Volume is going to be 1. Volume is going to be 125. And when you compare the surface area to volume ratio, all right, so when we do that, it's 6/1 versus 150/125. You get 6 versus 1.2, that number 6 is greater than 1.2. You want a large surface area to volume ratio because you want the cell needs to be able to feed the entire volume of the cell efficiently. So if you have a large cell, what I'm trying to say is that if you have a large cell, the surface area to volume ratio decreases. Because if you think about the equation, right, the volume is going to increase at a really fast rate, right, as the cell gets bigger and bigger. And so there's more and more volume. There's more and more of the cell. The volume within the cell, it's going to increase. And so this cell needs to be able to feed the entire volume of the cell, get rid of waste really quickly. And the larger the cell, the less efficient it is, and it's going to have a harder time to survive. And so cells are small because if they're small, the surface area is much larger than the volume inside. So there's a lot of surface area for food to come in, for waste to escape or leave the cell, and it's going to be much more efficient for the cell to be small. Okay, so this is why cells are small, because that larger surface area to volume ratio increases the rate of chemical exchange between the cell and the environment. And you know, we are multicellular. We're made up of many cells, and each cell is very small, which increases the surface area to volume ratio. Okay, so let's go over some really unique and amazing examples of how our body and how organisms increase surface area to make absorption of nutrients and getting rid of waste much more efficient. A good example of increasing surface area would be your small intestines. Okay, so within your gut, okay, this is what the small intestines are in peachy color, and then the large intestines is in gray. Okay, if you were to remove or measure, let's just say, measure your intestines, small and large together, because they're connected, it would be about 15 feet long, and it gets stored, you know, in your abdominal cavity. It's highly folded, okay? But when you look inside of the intestines, so this is like what it looks like on the inside of your intestines, it's very bumpy, okay? It's not smooth. Your intestines is not smooth. It's very bumpy, and it's highly folded. And see these like finger-like projections? Okay, this fold right here, it's called villi. And then when you take a microscope to the villi, each villi has tiny villi, which are called microvilli. And so the surface area of your intestines it's highly folded. The surface is highly folded, which increases surface area. And I know that in our textbook, it even says that the surface area of your intestines is so big that it's the size of a tennis court. Okay? So you have the surface area the size of a tennis court, and it makes it so efficient to absorb food and then to get actually, your intestine doesn't get rid of waste, but it absorbs food, okay? So this is like the villi, and then when you look at the villi, it even has microvilli, okay? So it's highly folded surface to increase the absorption of nutrients. And so the villi are the finger-like projections like this, and then the microvilli are the tiny projections on the villi, okay? And then when you look at plants, okay, plants, they have this root system that also increases surface area. Because if you had like a plant, okay, so let's just draw like a plant. This is supposed to be a flower, okay? And if it had just one root that went down to absorb water and nutrients, there's not a whole lot of surface area, right? Compared to a highly branched system, right? And then when you take a microscope and you look at the roots, you can see like these root hairs. And these root hairs are going to be extending outward into the soil like this to increase surface area even further, right? Because these plants, they need to increase surface area to absorb water and nutrients and minerals that the plant needs to survive, okay? So root hairs is, in plants, an example of increasing surface area to absorb water and minerals. Okay, now let's move on to the organelles found in eukaryotes. The nucleus, right? The nucleus, its function is to control, its function is to hold the DNA, okay? That's where the DNA is kept, and it's called the control center of the cell because the DNA has genes, and genes are going to code for proteins, and it's the proteins that do the work in the cell. So in a sense that it's like the control center of the cell. It contains our DNA, and like I mentioned earlier, there's a nuclear membrane or nuclear envelope. And when we say membrane, anytime we talk about a membrane in reference to living things in cells, it is a phospholipid bilayer. Okay, so when we say membrane, we're talking about phospholipid bilayer. And it has two phospholipid bilayers, so it's a double membrane. And it's continuous, meaning it's attached to the rough endoplasmic reticulum, which we'll be talking about. Endoplasmic reticulum, short, is ER. There are these little holes or pores. The nucleus has these little holes, and the holes are where like mRNA is going to exit the nucleus. This is where ribosomes are made in the nucleus, so they're going to exit the nucleus through the pores. And then chromatin. So when I say the DNA is kept in the nucleus, the DNA is going to be wrapped around proteins, and the DNA structure that's wrapped around proteins as a whole, we call it chromatin. Okay, chromatin. It's the complex of DNA and proteins. It makes up chromosomes when the cell is about to divide. But when you look inside the nucleus of a cell and you can see the DNA is going to be wrapped around proteins, and that whole complex is called chromatin. And the nucleolus, okay, the nucleolus is a structure or a region within the nucleus that's slightly darker. It's more compact and darker. That's where ribosomes are being made, okay? So here's a nice drawing of the nucleus. So this is the nucleus, and you can see those holes. Okay, those are the holes. Those holes, if you look closely, there are several protein complexes that make the hole, okay? But inside, there's the DNA wrapped around proteins, and we call that chromatin, okay? So DNA is like this blue strand, okay? Do you see that blue strand? And the blue strand is wrapped around these purple proteins, and those proteins are called histones. We're going to learn all about that when we go over DNA. But the proteins that the DNA wraps around are called histones, okay, or histone proteins. And all together, we call that chromatin. So all of that chromatin is in the nucleus. See this darker region in the nucleus? That's called the nucleolus. That's where ribosomes are being synthesized and made. Those ribosomes are going to exit through the pores and go attach themselves to either the rough ER, that's the rough ER, or they're going to be floating, ribosomes are going to be floating in the cytosol. Okay? So you can see the double membrane of the nucleus. You have an inner membrane and an outer membrane. The outer membrane is continuous. It's attached to the rough ER. Okay, so the rough ER is also an organelle. The nucleus is an organelle with a double membrane. The rough ER is also an organelle. We'll be talking about that very shortly. So those ribosomes that are made in the nucleolus, what do? They make proteins, okay? They make proteins. You have a large, so this is a ribosome down here, okay? You have a large subunit, and then you have a small subunit, okay? I like to say that the ribosome is like a hamburger bun, okay? Like if you go to McDonald's or wherever you get your burgers, okay? In and out, right? You've got the top bun, which is usually bigger in size, and then you have the lower bun, which is usually flatter. So ribosomes look sort of like that. You've got the top bun and the lower bun, the large subunit and the small subunit. And they come together. They come together in protein synthesis. But when they're not making proteins, they can come apart, okay? So what are ribosomes made up of? They're made up of two things, rRNA and protein. rRNA, the little r stands for ribosomal because they make up ribosomes. RNA, remember RNA is a type of nucleic acid. You've got the adenine, uracil, guanine, and cytosine. Okay, those are the four nitrogenous bases that make up the nucleotides in the nucleic acid RNA. Okay, and then proteins. So That's what ribosomes are made up of. I mentioned the large subunit and small subunit. So you're going to find ribosomes in two places, okay? Two ribosomes in the cell. Where are they going to make proteins? Some of them are going to just be floating in the cytosol, like free floating, and they produce proteins that are going to stay inside of the cell. Whereas the ribosomes that are bound to or attached to the rough ER, those proteins that are made on the rough ER, they're going to be exported out of the cell, okay? Usually they're going to be exported out of the cell. Like, for example, your hair and your nails, right? They're made out of protein. How did they get out of the cells? They get out of the cells because those proteins are going to be made on the rough ER, okay? So the rough ER with the attached ribosomes, you know, those proteins are made on the rough ER and they get shipped to the Golgi, and the Golgi is where those proteins are going to be modified, and then those proteins are going to be put in a little vesicle and then they get shipped out. This is what we call the endomembrane system. Endo meaning inside, okay? So like inside the cell, there's all these membranes and they're all somehow either connected, or the product, like the protein, moves through them, okay? It's like a conveyor belt where the protein that's made on the rough ER, it gets sent to the Golgi and then the Golgi ships it out. So they're connected. They're all part of this whole endomembrane system, okay? So more on that later. So the ER, or endoplasmic reticulum, what is it? It's basically a network of membranes and sacs. There's two types of an ER. You got the rough, and it's called rough because it literally has a rough appearance due to the ribosomes attached to it. Its job is to package proteins. The ribosomes are attached to it, so it's going to package the proteins for secretion. Secretion means it's going to release it from the cell. And it's going to transport the protein to the Golgi. The protein made on the rough ER gets shipped to the Golgi in a little vesicle. And also sometimes, it replaces the proteins that are made on the rough ER. It replaces the proteins that are going to take part or are part of the cell membrane. And we're going to be talking about the cell membrane in another chapter. But the cell membrane is made up of phospholipids and proteins. And some of those proteins that are made on the rough ER are going to be part of the cell membrane. Okay? All right. And then the smooth ER, they don't have any ribosomes on the surface, so they have a smooth appearance. And their function is not to make proteins, but this is where they're going to synthesize lipids. This is where you metabolize carbohydrates, you detox drugs and poisons, and also store the calcium ions. Okay, so the rough ER does one thing, right? That's where proteins are made, and the rough ER is going to transport the proteins to the Golgi. They have that one job related to proteins. Whereas smooth ER does so many things. Okay, they do a lot of things. They make lipids. They help metabolize carbohydrates. This is where it really depends on what part of the body we're talking about. Like, for example, in the liver cells, there's a lot of smooth ER because the liver detoxes drugs and poisons. Okay, so like usually when somebody is an alcoholic, they might have liver failure because alcohol is a poison, it's a toxin, and that's where alcohol is metabolized. Okay, so somebody that's an alcoholic might sadly die of liver failure, okay, or cirrhosis. And then like muscle cells, your muscle cells the smooth ER stores calcium ions because calcium ions are necessary for muscle contraction. Okay, so really the smooth ER has a lot of jobs and functions, but it also depends on what cells we're talking about within your body. Liver cells, the smooth ER is going to detox drugs and poisons. Muscle cells, the smooth ER is going to store calcium ions, etc. Okay. So this is what the rough ER looks like. It's very bumpy. It's got the ribosomes attached to it. The ribosomes are where the proteins are made. The proteins that are made on the rough ER, they get shipped in these little vesicles, and they're going to go to the Golgi. Okay, they're like little vehicles, little Ubers, or what's the other? Yeah, I forget the other one. Lyft, right? Uber and Lyft. They're going to ship them to the Golgi. Oh, I forgot that this was in the presentation. So a few years ago, okay, at the Met Gala, right? Beyonce, she wore this dress, and I remember on Twitter, like people were like, "Oh, she looks like she's the rough ER." Okay, like it looks like membrane, right? And then all those pearls and jewels look like ribosomes. So, like textbook, looks just like her dress. Okay, so the Golgi. The Golgi receives materials from the rough and the smooth ER. Okay, so what does the Golgi do? Its function is to synthesize and package materials for transport in vesicles, and they're going to produce the lysosomes, okay, depending on what they receive. So let me go over that right now. Okay, so here's the Golgi. It can receive from the rough ER, so rough ER, it can receive proteins, and within the Golgi, within this the stack of membranes, they can be modified. They could add sugars to it, like they can add sugars to it. They can fold the protein further, modify the protein, and then here is the protein inside of the little vesicle that gets shipped out, and it can become a lysosome. Okay, so in this case, we're going to be talking about lysosomes too, but lysosomes are organelles with enzymes, which are proteins, inside a vesicle that's going to actually stay in the cell, or you could have a vesicle with proteins that are going to get shipped out of the cell, so going to get shipped out. Okay, but if the Golgi receives lipids, so I'll just put L, from the smooth ER, okay, it gets modified in the Golgi, and then it can get shipped out, or actually could become part of the cell membrane. Like maybe it's making phospholipids. So, you know, the smooth ER could be making phospholipids, and then the Golgi is going to modify it and then send it to the cell membrane. Or, you know, other lipids like steroids and waxes, okay, they're going to be made in the smooth ER. So the Golgi, you have the cis phase that receives it from the rough or the smooth ER, okay? And then the trans phase is where it's leaving, okay? All the materials that are modified in the Golgi, when they get shipped out in these vesicles, the side that is shipped out, that's where we call it trans. So you enter in through the cis phase, and then you exit out through the trans phase. All right, so the lysosomes, like I said, here, you know, the proteins that are made on the rough ER, they get modified in the Golgi, and they get shipped in a vesicle, and they might stay inside the cell, and those proteins are enzymes, and that becomes an organelle, and that organelle is called a lysosome. Lysosomes have enzymes that are going to be used for intracellular digestion. Intracellular, like means inside the cell. There's going to be digestion that's happening. And then also there might be materials within the cell that need to be recycled. And then there's also what we call programmed cell death or apoptosis. And we'll be talking about that in just a sec, okay? So what's inside of lysosomes? Proteins called enzymes, okay? And they're called hydrolytic enzymes. We learned in the previous chapters that hydrolysis, right? You should remember what hydrolysis is. It's using water to break down macromolecules, right? So hydrolytic enzymes, these are enzymes that break down large molecules, okay? So in this picture here, there's food coming into a little vesicle, and then we call that a food vacuole. Here's our lysosome with the digestive enzymes. The membrane of, again, when I say membrane, you should think it's a phospholipid bilayer, okay? The membrane of the lysosome is going to fuse with the food vesicle or food vacuole, and now those enzymes are going to start breaking down that food. So now you have food into smaller pieces, which can be digested by that cell. And then if things need to be recycled within the cell, let's say that there is a mitochondria that's like really old, okay? The lysosome is going to fuse with the vesicle that contains the mitochondria, and it's going to digest the mitochondria, okay? And then, so that's why we may recycle cells materials. Like if there's like old organelles, the organelles can be broken down and the parts can be reused, okay? And then let's have a programmed cell death. So apoptosis is when it's basically called cell suicide. And that sounds kind of crazy, but it's actually very important with development, okay? You know, when you were developing within your mother's womb, you know, like, for example, your hands and your feet, you know, there was a lot of skin that connected your fingers and toes together. They were webbed, okay? Your fingers were webbed. But what happened to those skin cells? Those skin cells had to die. Your body had to get rid of them. And the only way to get rid of them is through apoptosis, lysosomes. So what happens here are the lysosomes, they start to eat the cell from the inside out, okay? And so the cell dies because the lysosome is going around and digesting all of the organelles and everything all the parts of the cell is being digested by the lysosomes, and the cell will just die, okay? And so that's really, apoptosis is actually very important. Oh, another example why apoptosis is important is because when a cell has a mutation, like let's say during cell, DNA replication, when DNA is being copied, let's say there's a mistake, and the cell, there's a signal that says, okay, you do not want to divide. You don't want to make more cells with the same mistake. So there's a cell signal that causes that cell to die by apoptosis, okay? So cancerous cells or cells with mutations, DNA mistakes, those cells are going to commit cell suicide so that the whole organism stays healthy, okay? So just as a quick review again, the endomembrane system, what is it? It's the nucleus. It includes a nucleus because it's connected to the rough ER. So inside the cell, there are all these membranes that are either connected or they are not directly connected, but they're related because the rough ER and the smooth ER, they send what they're making to the Golgi. So the Golgi is also part of the endomembrane system. And then perhaps whatever's being made in the Golgi stays in the cell, like this would be the lysosome. So lysosomes are also part of the endomembrane system. So endomembrane system is nucleus, rough, smooth ER, Golgi, lysosome. Okay, and then this picture you can't really see because of me, okay? But that's the proteins that are going to be secreted out of the cell. Those are proteins that are made in the cell that need to be secreted. Okay, vacuoles. There was a food vacuole that you saw earlier. So vacuoles are basically little vesicles that can store things like food, water, minerals, and these are more for like plants. Vacuoles in plants are going to store water, minerals, pigments. So when you look at a flower, when you look at a flower, like the petals of a flower, like a rose, think of a red rose, you can see the petals as being red because the red pigments are being stored within the vacuoles, within the cells of the petals. And then also plants can be very poisonous and they're usually stored within the vacuole. So these are membrane-bound vesicles. Just examples, food vacuoles, contractile vacuoles. Contractile vacuoles we'll talk about in the next, or no, when we go over the cell membrane, which is the next chapter in our textbook. Those are found in paramecium, and these are vacuoles that like pump out water because water is coming into these organisms and then they need to pump it back out. But plants have a very large vacuole, one large central vacuole. So this is a plant cell right here. And then the largest organelle that you can see inside of the plant cell is the central vacuole. That's where water is stored. Some ions are stored, and then, like I said, depending on what part of the plant we're talking about, it could be pigments, it could be poisons, okay? But you can see here that this central vacuole, it's filled with water and ions, okay? And then this is the nucleus right here. The chromatin is inside, and then you also see 1, 2, 3. I see 3 chloroplasts, which are going to be responsible for photosynthesis. In comparison, animal cells, we do not have a large central vacuole. We have like little small vesicles or vacuoles that, you know, could be food vacuoles or vesicles, okay? I don't actually see any in this picture. I mean, it could be, no, that's a lysosome. Yeah, I don't actually see any in this picture. Okay, and here's a diagram of a plant cell, right? You have the large central vacuole with chloroplasts, mitochondria, Golgi, rough, and smooth ER. Let's go back to this one. Yeah, so you could see the endomembrane system, nucleus, rough ER, smooth ER, Golgi. All right, let's talk about the mitochondria. You guys, I mean, everybody has heard of mitochondria. It's like it's reached meme status. And when I say meme status, it's like a joke now, right? Mitochondria, the powerhouse of the cell. Well, you know what? In AP biology, that's not good enough. You can't say powerhouse of the cell. Please do not say that on the exam in a free response. What you're going to say is that it is the site of cellular respiration. And we're going to be talking about cellular respiration in chapter 9, okay? Its job is to make energy, but what is that energy in the form of? ATP, okay? ATP is the molecule that provides energy for our cells. Mitochondria, what is its structure like? It has actually 2 membranes. When I say membrane, you should think phospholipid bilayer. So there's 2 phospholipid bilayers. There's an outer and inner membrane. Okay, so you can see that here. There's an outer membrane on the outside, and then you have an inner membrane. And the thing about the inner membrane, it's highly folded, whereas the outer membrane is pretty smooth. Okay, the inner membrane is highly folded, and there's a reason for that, right? When we have a highly folded structure, that increases surface area, okay? And when we increase surface area in the mitochondria, that's going to allow for more ATP to be made, okay? So those folds are called cristae, okay? See these folds? These folds are called cristae, the folds of the inner membrane. And the cristae, it contains these enzymes that are going to be responsible for ATP production. So the more folds you have, the more ATP is going to be made, right? Because it increases surface area to increase ATP synthesis. And then within the inner membrane, that fluid, we don't call it cytosol, we call it the matrix. Okay? All right, yeah. And then chloroplast. Chloroplast is the site of photosynthesis. It too has a double membrane. You have an outer membrane and an inner membrane. The inner membrane is not highly folded because that's actually not where photosynthesis is taking place. Photosynthesis takes place on these thylakoids, and these thylakoids also have a membrane. These little discs, okay, see these little discs? They look sort of like flat M&Ms, okay? Those are the thylakoids. The membrane, surface of the membrane of the thylakoids is where it's absorbing solar energy to make food for the plant. So when we say chloroplast is a site of photosynthesis, like photo means light, synthesis it means to make, where the plants are using light to make food, right? Chloroplast is the organelle that makes food using light for plants. So the thylakoid, those little discs, like those little M&Ms, the stack itself is called grana. Okay, so right over here, granum, okay, is going to be singular. Grana is, like I wrote here, is plural. So this is one stack, granum, many stacks, grana. And then there's fluid. See, there's fluid that surrounds, I'm talking about inside. We're inside the chloroplast right now. There's fluid inside the chloroplast, and that's called stroma. The thing about I said that the thylakoid membrane is where the light is being absorbed, and it's because there are pigments. There's pigments embedded in the thylakoid membrane called chlorophyll. Okay, pigments are basically molecules that capture light. Okay. Okay, you definitely need to know about this, okay? You need to know about endosymbiosis or endosymbiont theory, okay? It's a theory that says that mitochondria and chloroplasts, they once were little prokaryotes or like bacteria. They were little prokaryotic bacterial-like cells, and they were eaten, okay? They were eaten by a larger cell. And instead of being digested by the larger cell, they stayed alive inside of the larger cell. And endo means inside, symbiosis or symbiont means that they're working together. Okay, they're living in close association, in this case, inside of the larger cell, and they're like surviving together, living together. So you can see here, the mitochondria is now, it becomes an organelle. Instead of being a separate organism, over time it's become part of the larger cell, okay? And we think that mitochondria were taken in first, okay? And then chloroplasts, the prokaryotic chloroplast was taken in later, okay? So that's the endosymbiont theory, and there is evidence for that. Let's go over the evidence. So mitochondria and chloroplasts share similar origin, meaning like they used to be prokaryotes that were taken in by a larger cell. So prokaryotic cells engulfed, we think they were engulfed, meaning swallowed in, by ancestors of eukaryotic cells. And the evidence is that both mitochondria and chloroplasts have two membranes. They have two membranes. So how is that evidence? Okay, let me go ahead and draw a cell. So let's say this is our larger cell, okay? Swallowing up the mitochondria, the precursor to mitochondria. The mitochondria, like let's say just the prokaryote that becomes mitochondria. And then when it gets swallowed in, it takes in, let me erase that. It's a little confusing. It's taken in the membrane. Okay, see the membrane? Surrounds the little mitochondria and it gets pinched off, okay? The membrane of the larger cell pinches off or it takes in the mitochondria, and now do you guys see how that little mitochondria is now in a vesicle? Okay, that little mitochondria is now in the vesicle, and this becomes the outer membrane of the mitochondria, and then the original membrane of the mitochondria is the inner membrane. And same goes with the chloroplast. Okay, that little chloroplast precursor prokaryotic cell is taken in, the large cell brings it in a vesicle, and you have an outer membrane and inner membrane. So that's one of the evidence that we think prokaryotes or eukaryotes evolved, because they took in precursor cells and endosymbiosis. They, instead of being digested, they stayed inside of the larger cell, and they became organelles, mitochondria, and chloroplasts. In addition to having a double membrane, they have their own ribosomes, okay? So chloroplast and mitochondria, they have separate ribosomes. Let me just draw really quickly. So this is the mitochondria. Remember I said that the outer membrane is smooth and the inner membrane is highly folded. The folds are called cristae. Well, inside the matrix are mitochondria are ribosomes. And these ribosomes do not resemble the ribosomes that are outside of the mitochondria. So within a eukaryotic cell, you have ribosomes. Remember, the ribosomes could either be free-floating or attached to the rough ER. Those ribosomes are completely different than the ribosomes that are found in the mitochondria. In fact, when you compare ribosomes in the mitochondria with bacteria, ribosomes within bacteria, they're very similar, okay? Same with chloroplasts. Chloroplasts and where should I draw that? I'll just draw the chloroplast here. So you have the double membrane. And then you have, these are the thylakoids, those little flat M&Ms, okay? You have ribosomes that are just kind of floating in the stroma of the chloroplast. And they too are nothing like the ribosomes that are on the outside of the chloroplast. They're much more, the sequence of the RNA within the ribosomes are much more similar to the RNA sequence found in bacteria. And then also within a cell, mitochondria can divide and reproduce very similarly the way that bacteria do. Same with chloroplasts. They reproduce within the cell, just like bacteria do. Okay, now let's talk about peroxisomes. These are organelles that break down fatty acids. So when you eat fats, within the cell, the fatty acids get broken down for respiration. You use it to make energy, and also the peroxisomes are there to detox alcohol. So you're going to find a lot of peroxisomes in liver cells, okay? And it's also involved in production of hydrogen peroxide. Cytoskeleton, this is an excuse me, like cytoskeleton refers to inside of the cytoplasm, inside the cell, you have a skeletal network that's going to provide structure for the cell, okay? It's going to provide support, that's what I mean by structure. Motility, and we're going to talk about that because when we get to talking about flagella and cilia, it's made out of cytoskeleton protein fibers. And they also regulate biochemical activities. So this is a beautiful photo where they have, using fluorescent molecules, they have, what is it called? Labeled, okay? Fluorescently labeled the cytoskeleton. And you can see it in green, you can see it in orange, okay? It provides support for the cell. So there's 3 types of cytoskeleton fibers, and actually you don't need to know all of this stuff, okay? You know, let me just highlight the ones that you should know. Like for the microtubules, these are the largest fibers. They provide support for the cell, but, oh, and then also the organelles, like let's say mitochondria. If mitochondria are going to move across the cell, like let's say that a cell is like a room, it needs to go from one side of the room to the other side of the room. The mitochondria, using these motor proteins, the mitochondria is going to move along the cytoskeleton. So along the microtubule. So let's say that this is a cell, here's a mitochondria, and then it's going to travel along, let's say this is a microtubule, using these like motor proteins. They're kind of like little feet. The mitochondria is going to move along. It's kind of cute. There's like videos online if you can see it. Oh, I apologize, but that's not the best picture. But the motor proteins, like a highway, like a road, they move the mitochondria along the microtubule. Okay? Okay, and then microtubules, definitely you guys need to know this. They form the spindle fiber for mitosis and meiosis. Meaning that in cell division, mitosis and meiosis, you're going to see that the chromosomes, they need to move into the appropriate cell, and the only way they can get into the chromosomes, can get into the appropriate cell, is by the microtubules. It's the microtubules that push and pull the chromosomes into the right cell. Okay? And then the microtubules are also the component of cilia and flagella. You know how I said, oh, you don't need to know everything? It seems like for microtubules, you do need to know all of this stuff, okay? I think microfilaments and intermediate filaments are not as important as the microtubules, okay? So there's 3 types of cytoskeleton fibers: microtubules, which are the most important, and then you have the microfilaments, and then you have the intermediate filaments, okay? Yeah, I don't think that you need to know too much about the microfilaments and the intermediate filaments. Just know that the cytoskeleton that provides support for the cell provides, in this case, the microtubules not only provide support, but also organelle movement and forms a spindle for mitosis, meiosis. And then we're going to talk about the microtubules in terms of components, or they make up cilia and flagella, okay? Okay, so centrosome, the centrosome is in animal cells, okay? It's in animal cells in which microtubules grow. Remember how I said microtubules are going to be really important for moving chromosomes during mitosis and meiosis? So mitosis, meiosis, those microtubules, where do they come from? They come from the centrosome, okay? The centrosome. And so the centrosome has two centrioles, one, two. There's two centrioles that make up a centrosome, and when you zoom in on a centriole, you can see that the centriole is made up of microtubules. Okay, I can count 1, 2, 3 microtubules, 1, 2, 3, 1, 2, 3. So there are 9 sets. There's 9 sets of 3 microtubules that make up a centriole, and there's 2 centrioles that make up a centrosome. Okay. And from the centrioles, these microtubules are going to radiate out, grow from, and they're going to be very important in grabbing and moving chromosomes around the cell during mitosis and meiosis. Okay, so the centrosome is made up of centrioles, and each centriole is made up of microtubules, and from the centrioles is where the microtubules grow from during mitosis and meiosis, and they're going to help move chromosomes around during cell division. So now let's talk about cilia and flagella, okay? Cilia and flagella are also made up of microtubules, but their purpose is not to move DNA around, okay? DNA around during mitosis, meiosis. It's not to provide cellular structure, not to provide cellular structure and support. Cilia and flagella is for movement. Okay, it's for movement of the cell or the organism. Flagella, let's talk about flagella first. So sperm, okay, sperm, right? When you guys all know what sperm does. They fertilize the egg. But how do they get to the egg, right? How do they get to the egg in the fallopian tube, the organ within the female body? How does it get to the egg? It swims using the flagella. Flagella, most sperm only have one flagella, so that's what I mean by few. There's usually just one, and it's usually long in comparison to the cell. Okay, and compared to cilia, you're gonna find one of them, and it's gonna be pretty long. It helps the cell or the sperm to propel through fluid. Okay, through water or fluid. And then cilia, they're pretty short in comparison to flagella, and you're gonna find a lot of them on the cell, okay? And the only example that, actually there's a couple of examples, okay? But let's go over one example, which is a paramecium, okay? So there is a single cell organism, mecium, that's supposed to be a U-M, paramecium. Okay, paramecium, they're single cell organisms. They're a type of protist that lives in like lakes and streams and fresh water. Okay, they're covered by these short little cilia, and the cilia will beat. They're like little, I guess you could say they're like little oars that kind of move the little organism through the water. So all the cilia will like move the organism through the water, okay? And so there's a lot of them. There's like hundreds of cilia. This little paramecium is covered. It's basically like a hairy little organism covered by cilia. The cilia are moving the organism through the water, okay? And so it's used for locomotion. Locomotion means to move. Okay, and both flagella and cilia have this 9+2 pattern, meaning there's 9 pairs of microtubules. You can count them. And then there's 2 microtubules in the middle. So there's 9 pairs, and then there's 2 in the middle. So if you were to like cut open a flagella or a cilia, you would find microtubules in this pattern. Oh, the other example of cilia that humans, we also have cilia. Besides humans, having sperm, humans also have cilia. Cilia is found in our trachea, okay? The trachea is part of our respiratory tract that leads to this, the trachea that leads to our lungs, and the cilia are constantly beating in an upward motion. And what it's doing is it's moving mucus and anything that gets caught in the mucus upward, okay? Because you don't want dust and things to get down into your lungs. So the mucus is there to capture, it's like the sticky substance. It captures dust and bacteria, viruses, and then the cilia are constantly beating in an upward direction, moving the mucus out away from your lungs into your nasal cavity, your throat, and then you might spit it out or blow your nose and all of that mucus comes out, okay? Okay, and then on the outside of all of your cells, you have the ECM, or the extracellular matrix. So this is our phospholipid bilayer, or the cell membrane, right? And outside is the ECM, extracellular matrix. There's proteins like collagen. There's there's collagen, and then this green stuff, okay? Those are the glycoproteins, or proteoglycan complex, meaning it's like proteins and sugars together. And that causes it to be very sticky so that cells can stick together. And the whole function of the ECM is to strengthen tissues because they're like sticking together, but also molecules in the ECM are going to be used to transmit signals, okay? Cells can like communicate with each other through the ECM. Molecules can from one cell can come in through the ECM, extracellular matrix, and communicate with the cell next to it, okay? Okay, and I believe, I was going to say last topic, but we're almost done, okay? Or inter, not intra, because intra means inside the cell. Inter means between the cell, okay? Like we're talking about between different cells, like how are they connected? Okay, so intercellular junctions, and this is only in animal cells. Like if you have one cell here, that's one cell, and then a second cell here, the junction is where they meet. Okay, junction is like where they meet. So now we're talking about what's happening between these two cells. What connects them? What connects these two cells? What's the intercellular junction between these two cells? Well, first of all, let's talk about tight junctions. These two cells are connected via tight junction, and that's what's going on here. You can think of tight junction as being like threads that connect the two cells together, okay? So, I mean, when you look at your cells or your skin cells, I mean, you have actually multiple layers of skin cells, and how are they connected? One way that they're connected is by the tight junction. Two cells are fused to form a watertight seal, meaning that it's like threaded together so that nothing can like go in between them. Okay, like water, it's like tight. It's really tight. Nothing can go through in between the two cells. Next, you have the desmosomes, which is over here. Desmosomes are like nails that rivet, nails or rivets that like connect the two cells together, fasten the cells together, okay? Same idea with the tight junction. You guys, the whole purpose of tight junction and desmosome is to really make the two cells connected, glued together really tightly. And then gap junctions is different though, okay? Gap junctions has a different purpose. The purpose of the gap junction are they're basically proteins that are like little tunnel system proteins or channels in which molecules can go through, like ions can go through them. So ions can go from one cell to the other cell. They're like little tunnels, okay? Like small molecules can pass. All right, so plant cells, there are some things that are not found in animal cells, like we do not have cell walls. Plant cells have cell walls. And you can see in this picture, like this is the cell membrane, and then on the outside of the cell membrane is a thick cell wall. And plants have two types of cell walls. You have the secondary cell wall and then the primary cell wall. You don't really need to know that anymore. Just know that it's like a cell wall. Okay, plants have cell walls, and the cell wall is there to protect the plant and to maintain its shape. It's made out of cellulose. Remember, it's made out of cellulose. Actually, maybe I have it on this slide here. Yeah, it's composed of cellulose. Remember, cellulose is a polysaccharide that is not digestible to animals. It's a polysaccharide made out of beta glucose, right? Which means that it's not going to be digestible to animals. And then this is sort of like gap junctions in animals. They have plasmodesmatas, which are like little channels between plant cells, neighboring cells, so that ions can pass through, okay? So let's compare what's in plant cells only versus animal cells only. In plant cells, they have central vacuoles, right? So if I draw a plant cell, there's the cell wall, and then there's a cell membrane. Cell membrane is on the inside, cell wall is on the outside. The largest organelle in a plant cell is going to be the central vacuole that stores water. Chloroplasts also in plant cells, not in animal cells. They have the cell wall, and then they have plasmodesmatas, which are a lot like gap junctions, okay? Animal cells only, we have lysosomes, plant cells do not. We have centrioles, plant cells do not. We have cilia, flagella, which plant cells do not. We have desmosomes, tight and gap junctions, and then we also have an extracellular matrix, okay? So those are some differences between animals and plant cells, and I think that might be it, okay? So that is the end of our presentation. I hope this was helpful. I hope this wasn't too long. All right, go have a really nice day. Talk to you later. Bye.